Good morning. My name is Minjae. I'm one of the MD-PhD students currently on neurosurgery clerkships here at Penn. Today I'm really excited to share my capstone projects, giving an overview of the surgical targets to treat movement disorders in functional neurosurgery, and hopefully shed some light on the mechanisms behind these therapies through some of the ongoing research projects that I've been involved in while working with my mentors, Dr. Halpern and Dr. Cajigas. Um, in functional and stereotactic neurosurgery, we treat a large variety of movement disorders, including dystonia, Tourette, spasticity. But the two most disorders we treat most commonly are Parkinson's disease and essential tremor. So I'll spend the next couple of minutes talking about the disease etiology, prevalence, and treatment options across the next several slides. For ET and PD, the two procedures that are really offered to patients are DBS and HIFU. DBS, deep brain stimulation, delivers this high frequency electrical stimulation to the deep structures involved in motor control, called the basal ganglia. We're able to achieve this by implanting electrodes, maybe one or maybe two. Whereas for HIFU, the high intensity frequency ultrasound, um, uh, uses these external acoustic beams that are focused in similar basal ganglia regions by using hundreds of smaller transducer elements, and this causes a permanent lesion in the brain. So DBS works by using, again, high frequency stimulation that can functionally inhibit the cells and essentially cause reversible lesions, whereas HIFU causes a permanent lasting lesion to achieve this therapeutic effect. Um, so going back to Parkinson's disease, so PD is actually a very prevalent, affecting 1% of the US population, and mostly more common in patients with 65 years and older. And its etiology involves the accumulation of toxic misfolded protein called the alpha-synuclein in part of the brain called the substantia nigra. This is a part of the brain that produces dopamine, which is crucial for motor control. In PD, this global depletion of dopamine can be observed across the brain, as shown in the bottom left. And because this dopaminergic deficits, the canonical motor symptoms seen in Parkinson's involve resting tremor, rigidity, bradykinesia, and freezing of gait. On the video on the left shows that the patients experience some feeling and some shuffling of gait, as we see in Parkinson's. And on the right, this video, hopefully we can appreciate that there is this decrement of the frequency of the motor movement, as well as the slowness and the loss of amplitude. Um, so the for movement is controlled by a series of circuitry that connects the cortex and the basal ganglia and the thalamus in a series of loops. In Parkinson's, we have this dysfunctional nigra causing a deficit in dopamine, and this causes this global dysfunction in this motor control loop. But essentially, we can actually hijack this circuitry by selectively modulating the activity in individual downstream nodes in each nuclei. So the common targets for HIFU and DBS is the SDN, the GPI, and then the VIM. VIM is actually mostly commonly used for tremor-dominant Parkinson's. So for essential tremor, it's the most prevalent movement disorders out there. It can cause debilitating action and postural tremor, as we can see in this video, and this really deteriorates the quality of lives for those patients. Unlike Parkinson's, we still don't know the exact pathophysiology of essential tremor, but we know it involves this abnormal activity across this circuitry between the dentate nucleus and the cerebellum, red nucleus, the VIM, the ventral intermediate nucleus of the thalamus, and then the motor cortex. And similar to BD, the VIM is the surgical target for HIFU DBS to treat these disorders and help patients. As for the clinical efficacy of HIFU and DBS, they have been shown to be highly effective. In large clinical studies, ST-DBS have shown to be effective in significantly reducing the severity of motor symptoms in Parkinson's, using this rating called UPDRS. And similar was the case for the VIM HIFU with a lasting effect. And while DBS and HIFU is a clinically effective therapy that is rarely offered across numerous comprehensive movement disorder centers across the world, there still exists critical unmet needs for patients and the caregiver providers. For example, the subset of patients develop the lasting postoperative adverse effects, such as visual impairment, gait, sensory, speech, well, depending on surgical sites. And furthermore, while most patients respond readily to these therapies, some patients experience suboptimal benefits requiring extensive programming of DBS configurations or even requiring a repeat surgery with their HIFU. And to answer both of these questions and address this clinical need, we need to have a deeper and better understanding of the mechanisms of how the therapies works and how creating a reversible lesions with DBS or irreversible permanent lesion with a HIFU affects not only the local surgical sites but influence the larger circuitry that these nuclei our surgical targets are part of. So I'll spend the next rest of the talk describing two of my research projects that I have been part of in hopes of answering these questions. So the first study that I like to showcase is characterizing tremor sweet spots and side effects or spots following thalamic high-intensity focused ultrasound in ET patients. So this is a study that I work with Dr. Halpern and Dr. Cajigas. I'm trying to understand, how does different side effects or tremor benefits are evoked based on selective ablations across the VIM thalamus in this patient population. So we looked into 42 patients who underwent VM HIFU, actually here at Penn. And for each patient, we segmented their sonication lesions from their postoperative MRI scan. In parallel, we assigned whether this patient had any side effect across the 5 different categories shown in below, and as well as whether they had a complete tremor resolution or some residual tremor postoperatively. And then we calculated a voxel-wise positive predictive map. This is basically a map that maps the probability of a given side effect occurring when that part of the brain has been sonicated. Across the 42 patients, the mean age of our patients were about 72, and then the rate of the postoperative side effect occurrences were actually quite comparable to that published in previous literatures. This shows the distributions of the sonications made across all the patients, with most sonications targeted and localized to the VM thalamus. And then when you look at the spatial patterns in this probability map, hopefully we can appreciate that the sonication regions causing side effects can be overlapping, but also quite distinctive across the side effect subtypes, as well as for tremor benefits. And next, what we did is that we want to test whether these maps have any utility in predicting whether arbitrary patients are likely to develop a certain side effect or not. So we took the individual sonication volumes, and based on voxelized probability calculations from the maps we produced, you were able to predict the side effect and tremor resolution occurrences with a higher predictive capacity of more than 87%, albeit this was the intra-cohort validation. So we have essentially this tremor C spot you want with sonication, and we have this side effect source spot you want to avoid. But we're then interested to understand how does this region have any biological basis that results in this clinical presentation. To answer this question, we looked at any intersecting white matter bundles defined based on tractography results passing through the sweet spots and sour spots. And we found that these spots occupy a distinctive white matter connections that pass the VIM in different subregions. So hopefully we can see the gray outline shown over here. So that denotes the VIM nuclei. And then we see that the speech and discoordination, hotspots are located more anteriorly, following this DRTT, the dendrothalamic tract. And then for the numbness and weakness, the hotspots are located more posteriorly, lying at the medial lemniscus. And then for tremor sweet spots, were also more posterior, but more superior, lying at this non-decussating DRTT. And hopefully in this video, we can see the intersecting sweet spots and then the sour spots occupying different white matter tracts in the vicinity of the VIM and its subregions. And based on these findings, we want to take the individual probability maps for the different side effects and term resolution. We want to kind of combine it into one single map where it can hopefully inform clinicians and surgeons to understand where's the area that is most beneficial for tremor, at the same time, area that is minimal side effect occurrences. And then we found that this area was localized to the right, to the posterior, to the VIM, again intersecting at the non-decussating DRTC tract as shown in this figure. And finally, we want to kind of put this our map into a clinical use. So we have this patient with ET planned for VM HIFU, and then we imported this tremor prediction map shown in the previous figure. This one has a black cavity, shown over here in the axial and corneal slices, and then relative to VIM, with the outline shown in this pink this vicinity. And then during surgery, our actual sonication was targeted as this circular outline right on top of the prediction map. And then postoperatively, this patient had a full term resolution without any side effects. So we're currently actively working towards building this more prospective study cohort and perform this out-of-sample cross validation. I'm really excited to share the results in our upcoming publication. So in summary, in this study, we found that the sonication areas that cause various side effect profiles occupy not only the different sub-regions within the VIM, but also these regions were connected to the white matter tract that relays to more distal neural substrates like cerebellum or the motor cortex. Hopefully, this can share some mechanisms that drive the VIM high through clinical benefits and explain why some patients develop decided on an adverse effects. Um, so this study was about HIFU. So I'll spend the next half of the talk talking about the second study that hopefully provides some understanding on the circular mechanisms of deep brain stimulation for Parkinson's patients. Um, so the second study was done with Dr. Cajigas and the lab members, where we worked on characterizing a microscale patient-specific motor cortex activity and networks following the clinically effective STN DBS. I mentioned that the ST-DBS is highly therapeutic to manage the canonical motor symptoms in Parkinson's, and we think that it works by modulating the both local SDN nuclear physiology and more distal cortex physiology by modulating the series of networks. But the precise mechanisms of how these circuitries engage are largely unknown and still under active investigation. So the question that we want to answer with this study was, how does SDN engage motor cortex physiology and networks to really achieve its clinical benefit? So we looked into three patients who were undergoing SDN DBS surgery, the Parkinson's patients. And then during surgery, what we did is we are able to intraoperatively stimulate different contacts of the electrodes, shown on these different levels, with variable stimulation parameters, testing different amplitudes. And based on each parameters, we can then use mathematical modeling to produce this electrical field distribution relative to your active contact. And based on that, we can then generate this volume of tissue activation of VTA based on biophysical assumptions. And then we have this atlas-defined rigidity sweet spot from a group of patients in a large clinical study. And then we then calculate this volumetric overlap between the individual VTA and this atlas-defined sweet spot. And then repeat that across different stimulation configurations as we go from more inferior contact to more superior contacts across the three patients. On the right, we show the locations of the three SDN electrodes shown relative to the SDN, shown in orange, and then the atlas-defined rigidity sweet spot located more posterolaterally and this nuclei. In parallel, we implanted this high density 1024 channel array developed by Princeton Neuroscience in this part of our brain motor cortex called the head knob, shown in this figure. And then from each channel, we're able to then calculate the changes of the spectral power across the three bands of interest, the low beta band, high beta band, and then the gamma band, relative to the baseline no stimulation condition. figure on the left, hopefully we saw the locations of the three cortical array across the three patients, as well as the SDN electrodes. And then on the right, hopefully we can appreciate this granular information we can capture using this high density array in the cortex. And this was during baseline and no stimulation. After usually the sweet spot stimulation, we found that the spatial patterns of low beta and high beta band were actually quite heterogeneous between the two subbands, but also across the three patients. On the other hand, we found that across three patients, that we found an elevated gamma band in this specific area in this more medial posterior part of the hand region. That's what we call the gamma hotspot, shown in the circle. And what we did next was we were interested to see where this hotspot was highly connected to in subcortical nodes involved in motor control. So we took individual channels and we used that as a seed in the resting state fMRI to generate the channel-specific functional connectivity map. And what we did next was we then correlated this individual voxels functional connectivity per channel with the actual changes of the gamma spectral power relative to baseline. Essentially producing the gamma band connectivity map. What this map shows in essence is the areas that is yellow. These are areas that are highly connected. So these are the regions where we see an increased in functional connectivity from the elevated gamma hotspot from the cortex. And then we see involvement of the bilateral motor cortices and some sonic subregions. And if you focused on a specific subcortical nodes, involving motor network, going back to the initial point of the talk, namely the striatum, the putamen, caudate, GPI, and SDN. What we found was that there's a functional connectivity to specific subregions of the individual subnuclei. So, for example, in striatum, it was mostly localized to the posterior lateral part of the nuclei, namely in the putamen. GPI and STN are more on the posterior lateral part of the nuclei, which is actually the DBS target for Parkinson's disease. And what we found in summary is that after the STN outlets defined rigidity sweet spot stimulation, the pattern of the beta band activity in the cortex was actually quite different across the patients, but it was the gamma band where we see an elevated activity in the specific area, that is more localized and focal in the medial posterior part of the hand knob. And lastly, we're able to take the cortex the cortical regions and then look back into connectivity into the basal ganglia subregions. And we found that the gamma hotspots were highly connected to the central hub of the motor network across the three striatum, GPN, and SDN. So far I talked about the functional neurosurgery in movement disorders, but what is the next frontier in this field? So beyond movement disorders, the field is actively moving towards treating patients with neuropsychiatric disorders, with a lot of research studies going on, both here at Penn and at different institutions. And then the goal is to really focus on understanding the neuroscience mechanisms, the circuitry, and the surgical targets across diverse indications. For example, at our lab with Dr. Halpern, we have been investigating ways to deliver more personalized stimulation strategies to treat OCD based on the electrophysiology biomarkers associated with the obsessive cues that are uniquely found per each patient. And another disease that the field is working on is understanding how to treat schizophrenia, such as auditory and verbal hallucinations. So recently, we wrote a paper below that is currently under review for journal publication, and understanding how we can deliver stimulation to the this area called substantia nigra reticulata, and see which connections, whole brain connections are engaged to achieve this clinical benefit and reduce the burden of hallucinations in this patient population. That's everything I have for today, and I just want to thank all my mentors and the lab members for these two projects. And if you have any questions about our work, please feel free to reach out. Thank you.